My name is Michael Pavelchek, and I'm from Austin, New York, and my research looked at asthma. So, roughly about 26 million people have been diagnosed with asthma in the US, according to the AFA in 2015. And the incidence of asthma and the mortality rates from asthma attacks have been on the rise over the last decade. So, there's two main types of asthma, allergic asthma, and that's an atopic disorder similar to things such as food allergies and atopic dermatitis, and non-allergic asthma, which is an autoimmune disorder, which is similar to things such as inflammatory bowel disease. So my study looked at allergic asthma, and allergic asthma is really characterized by this high affinity IgE antibody, which is its main inflammatory agent. And what's unique about this antibody is that it's produced through a process known as sequential switching. And sequential switching is this reorganization of amino acids, and this happens in these IgG1 memory B cells. And so these IgG1 memory B cells go through this process, and they become these high affinity IgE secreting plasma cells. Now, recently a study found that there's actually two subsets of these IgG1 memory B cells. A double positive subset, which expresses these two genes known as CD80 and CD73, and they differentiate into those high affinity IgE plasma cells. And then there's a double negative subset, which doesn't express those two genes, and they actually differentiate into these IgG1 germinal center cells. So really what my study looked at is previous research hadn't looked at the genes involved in that sequential switching process between those two different subsets. And so what I looked to see is what is responsible for that sequential switching process occurring in that double positive subset, but not that double negative subset. And so what I did is I did an RNA sequencing on those two different subsets to look at those different gene expression levels. And so what I ended up finding is that two genes, MYC and SWAP70, were upregulated within that double positive subset. And so then to verify this, I did a database profile this summer where I took Th2 and Th1 immune responses and once again compared those gene expression levels, and I was able to verify that those two genes, MYC and SWAP70, were also upregulated in that allergic immune response. And so what this means is that these two genes are really key to the generation of that high affinity IgE antibody, and they're key to that sequential switching process to occur. My study also looked at some of the inflammatory pathways. When I looked at this IgE inflammatory pathway, I actually found that it was upregulated within that double negative subset. So this actually indicates that we have an overproduction of not just high affinity IgE, but also low affinity IgE. And actually the low affinity IgE is actually being produced to a greater degree. So really what we can do with this, the applications of this, is current treatments for allergic asthma are pretty limited. We mainly have bronchodilators, which really just reduce the inflammation during an asthma attack. There's no real long-term treatments for allergic asthma. And so by looking at drugs such as Regeneron, which inhibits SWAP70, we could use this to prevent the production of that high affinity IgE antibody, and we could then reduce that inflammation during an allergic asthma immune response. And what's great about this is, as I mentioned, allergic asthma is similar to things such as food allergies and atopic dermatitis, in that they all share that same method of inflammation, that sequential switching process, and that high affinity IgE antibody, meaning we could this would not only help reduce that inflammation during allergic asthma, but could also apply to all these other different atopic disorders such as food allergies, allergic rhinitis, and even hay fever.